Yeah, it's right there. Well, yeah, my research project is focused on the identification of inverted and direct repeats that are involved in the formation of complex genomic rearrangements. So let's start with a brief introduction where I would like to point out the importance to understand the genomic architecture of the repetitive DNA sequences, as it is the 30% of the human genome, because based on their features like orientation, size, density, and distribution, it is possible to know what regions in the genome are prone to instability or more susceptible to rearrangement formation. As well, we can also identify if the formation of new structural variants is through DNA recombination mechanism, like, can you see my pointer? Well, never mind. Like non-allelic homologous recombination, where the recombination acts on substrates with a homology around 95% and a size between 5 to 10 kilobases. And based on the orientation of these repeats, we can have two different outputs. If it is between direct repeats, you can get deletions, duplications, or even amplifications. But if it is between inverted repeats, you can get inversions. It is also important to note that most of the non-allelic homologous recombination events result in several genomic disorders. As well, we can have more complex rearrangements through replication-based mechanisms like MMB or FOSTES, where they use different sets of inverted and direct repeats, resulting in a combination of structural variants, like this one. And this is the workflow that I follow to identify these sort of repeats. So we use the latest available human genome assemblies, HG19, HG38, and telomere to telomere, T2T, and then we run LAC for each one of in the 3 assemblies for each one of the chromosomes to identify the repeats. And we use this set of parameters. So we look for repeats with an identity of at least 80% and a minimum length of 200 base pairs. So then with a custom-made algorithm, we collapse all the repeats according to specific criteria. And after that, we made some basic analysis, creating bed files and calculating statistics, followed by annotation using refseq genes and repeats from repeat maskers. And finally, we analyze the genome-wide distribution of these repeats. So one of the main reasons why we use 3 different human genome assemblies was to analyze and compare the characteristics and distribution of genomic repeats in each one of the assemblies. I also want to highlight the previous work done using HG19 assembly, but using different set of parameters more tailored towards non-allelic homologous recombination substrates, where they use a minimum length of 1 kilobase with an identity of 90%. But now with further experimental observations, we already know that there are other mechanisms involved in the formation of complex rearrangements that use microhomologies or shorter repeats with a less identity. So that's one of the reasons why we're using less stringent parameters, because we want to compare the difference between the previous work done using HG19 and the work that we are doing right now with the new set of parameters. And we use the first complete gapless telomere to telomere assembly, as it presents advantages over the other 2 reference assemblies, because there were previously overseen regions that are now complete in T2T that include satellite arrays, ribosomal RNA genes, and segmental duplications, where we are expecting to find more repeats that were previously overseen. And for analyzing the repeats, we are using LAC to perform pairwise alignment of whole chromosomes to identify these repeats, and we develop an algorithm to collapse overlapping or nearby repeats in complex regions while still maintaining a homology above 80%. So this is a cartoon model to explain how it works the collapsing algorithm. So basically, we have 3 possible scenarios where we can collapse all the repeats. The first one of them is by overlap. So A and B are pair of repeats, and A1 and B2 are the following pair of repeats. And if they are overlapping, it means that we can try to collapse them, resulting in larger alignments. The next one is by distance. So if this set of repeats are at least 2 kilobases away from each other, it also means that we can try to collapse them. And the third one is a combination of both of them. So once we collapse all the repeats, we calculate the new homology between the new set of repeats, and we keep the collapses with homology above 80%. Otherwise, we treat the 2 repeats as separate. And this is an example of a real case scenario to observe how it's working the collapsing algorithm in real data. So in the picture that is at the right, you can observe in the lower panel that there are few repeats that are overlapping among each other. And in the upper panel, you can observe how they are already collapsed into one. And in the picture that is at the left, you can observe in the lower panel that there are several repeats that are not overlapping. And in the upper panel, you can observe how they are being collapsed by distance, resulting in larger alignments. So this is how it works, or collapsing algorithm. And once we collapse all the repeats, we take a look at the statistics, and we found that there are more inverted repeats in each one of the assemblies than direct repeats, and we also found that there are more direct and inverted repeats in T2T than in the reference assemblies. And as expected with the parameters that we initially set, the homology goes from 80% to 100% in the three assemblies, either for direct or inverted repeats, and the length goes from 200 base pairs to several million base pairs. And for example, this one right here that is located in T2T is very large compared with the other two reference assemblies. And this one is specifically located in the short arm of the chromosome 13, which it is one of the regions that was previously overseen in the reference assemblies. And then we take a look at the distribution of direct and inverted repeats among the human genome assemblies, and we found out that the distribution is very similar for each one of the chromosomes in the three assemblies. And there are a few cases where we're still finding more repeats, either direct or inverted, in the reference assemblies than in T2T. But when we take a look at the second half of the human genome, we realize that there is an increase of inverted and direct repeats in T2T acrocentric chromosomes. As you can observe right here, those that are in the red boxes are the acrocentric chromosomes, and there is an increase of repeats in the T2T acrocentric chromosomes. And this increase is located specifically in the short arms of the acrocentric chromosomes, as you can observe in the chromosomes that are in the branches boxes. So in most of the chromosomes, the genome-wide distribution of these repeats is very similar, but there is an increase in the acrocentric chromosomes. As well, we found an increase of direct and inverted repeats in the long arm of the chromosome Y. And then we also were interested in looking for the composition of repeated elements in direct and inverted repeats. And we found that the most common repeat elements are sign elements in each one of the chromosomes, followed by segmental duplications, long terminal repeats, and line elements. And the distribution is very similar among the human genome assemblies. We also look for the presence of direct and inverted repeats near dosage-sensitive genes, as it can indicate regions in the human genome that are more susceptible to rearrangements. And we found that the proportion of repeats that are overlapping with genes is very similar between direct and inverted repeats. And we also look for repeats that are flanking genes. And we found that the proportion is slightly larger for direct repeats than inverted repeats. And right here, there is an example of a pair of inverted repeats that is flanking a gene in a region with a complex genomic rearrangement. So this is an array of a patient with a duplication marked in red, followed by an inverted replication marked in blue, and followed by another duplication as well in red. Right here, this is the pair of inverted repeats that are involved in the formation of this triplication that is inverted. So the blue one means that it's in the positive strand, and the green one means that it is in the negative strand. So what I want to highlight here is that this is one of the useful applications of LAST-C, because now we are able to identify the set of repeats that are involved in this type of rearrangements, because before it was more complicated to get the sequence and the regions where these inverted repeats were located. And right here, there are a couple more examples of a complex genomic rearrangement with the same genomic organization. What I mean is they also have a duplication, triplication, followed by other duplication. What is important to note here that it's that inverted repeats, even at a distance, are involved in the instability of the human genome and contribute to local genomic rearrangements. As conclusions, we observe a similar distribution of direct and inverted repeats among the three assemblies. We also identify repeats in T2T acrocentric chromosomes previously overseen in the other reference assemblies, and we observe an overall increase of repeats in T2T. The next steps, we want to make tracks of direct and inverted repeats available in USCC browser, and we still need further analysis and study of these repeats that are to study the complex genomic rearrangements observed in patients. So I want to thank all the team at PNRI, Lee, and Chris for their incredible support during my research project. So thank you. I'm happy to take any questions.
Hi, Liz. Very nice talk. Thank you.
Thank you.
So I was wondering, so this is a self-assembly, no? So you use HG19 original reads to, and then look for the repeat using the assembly from HG19 and then the T2T. So I was wondering, are you going to start trying now with patient data to actually assess whether one assembly versus the other would be more useful to actually analyze patient data?
Yeah, that's the point. That would be like the next step, like to analyze patients using different assemblies to see what are the difference if we are able to identify the repeats that are involved in this type of rearrangements using T2T assembly or other assemblies. But yeah, that's the idea.
And then I have a question because T2T is one person.
Yeah.
So how did you correct for that biases?
We didn't correct for that biases. We already know that we know that it's homozygous assembly and it's basically European ancestry. So we know that there are some mistakes right there. So yeah, but yeah, definitely it would be useful using other complete assembly that it's more diverse like the other reference assemblies.
In particular, because we know there's more diversity in structural variation in other populations.
Yeah, in other populations. Yeah, so I think that's one of the reasons why we are still finding more repeats in some chromosomes than in T2T because, as you already said, yeah.
Okay. Thank you, Liz.
Thank you, Ale.